Krebs cycle. Organisms derive the majority of their energy from the Krebs cycle, also known as the TCA cycle. The Krebs cycle is an aerobic process consisting of 8 definite steps. In order to enter the Krebs cycle, pyruvate must first be converted into acetyl-CoA by pyruvate dehydrogenase complex found in the mitochondria. In the presence of oxygen, organisms are capable of using the Krebs cycle. The reason oxygen is required is because the NADH and FADH2 produced in the Krebs cycle are able to be oxidized in the electron transport chain, thus replenishing the supply of NADA+ and FAD. Acetyl-CoA enters the Krebs cycle when it is joined to oxaloacetate by citrate synthase to produce citrate. This process requires the input of water. Oxaloacetate is the final metabolite of the Krebs cycle, and it joins again to start the cycle over again, hence the name Krebs cycle. This is known as the committed step. Citrate is then converted into isocitrate by the enzyme aconitase. This is accomplished by the removal and addition of water to yield an isomer. Isocitrate is then converted into alpha-ketoglutarate by isocitrate dehydrogenase, the byproducts of which are NADH and CO2. Alpha-ketoglutarate is then converted into succinyl-CoA by alpha-ketoglutarate dehydrogenase. NADH and CO2 are once again produced. Succinyl-CoA is then converted into succinate by succinyl-CoA synthetase, which yields 1 ATP per succinyl-CoA. Succinate converts into fumarate by way of the enzyme succinate dehydrogenase, and FAD is reduced to FADH2, which is a prosthetic group of succinate dehydrogenase. Succinate dehydrogenase is a direct part of the ETC. It is also known as electron carrier 2. Fumarate is then converted to malate by hydration with the use of fumarates. Malate is then converted into oxaloacetate by malate dehydrogenase, the byproducts of which are NADH.